Hi there, welcome to BioWorld. First of all, my sincere apologies for having taken forever to come up with a new video. So today's video is all about enzymes. Now, if we have a photo album of all the scientists who have contributed to our knowledge of enzymes, these two individuals will come up most frequently. They are Fisher from the year 1890 and Koshland from the year 1959. Their contribution to the field of enzyme technology, or our knowledge of enzymes, is the models that explain enzyme specificity. Fisher introduced what is known as the lock and key model, whereas Koshland came up with the induced fit model. Now, my discussion today is connected to our STPM Semester 1, Chapter 4 syllabus. In the STPM Semester 1 syllabus, under the subtopic 4.2, Mechanism of Action and Kinetics, Part A, you are to be able to illustrate enzyme specificity using the induced fit, that is Koshland's model, and the lock and key, that is Fisher's model. When they say illustrate, that means you should be able to roughly draw the diagrams representing both these models. So I'll start with the original model that we still use, that is the Fisher's lock and key model. To explain Fisher's model, let me use this as an example of an enzyme. As you know, enzymes have very specific active sites. So according to Fisher, the substrate must have a complementary structure to the structure of the active site so that it can bind to form an enzyme substrate complex. Now I'll show you an example of a substrate that seems complementary, but when it binds, you see, it doesn't really make a very perfect fit. Therefore, it will not be able to form an enzyme substrate complex. But if we have, for example, another substrate here. Now, this substrate is complementary to the active site. So you can see when it binds, it forms a perfect fit. Therefore, an enzyme substrate complex will form and a product will be produced. So from this, we can make a few conclusions about Fisher's model. The conclusions are, let's gather some points about Fisher's lock and key model. Firstly, his model indicates that the active site of an enzyme is rigid. This is similar to what you find in the keyhole of a lock. You know very well that the keyhole is rigid, and there is only a specific key that can be inserted into the keyhole to unlock. So similarly, there is a specific substrate that will be able to bind to the enzyme. And of course, from the many keys that we may have, one key will fit into the keyhole because it will have a complementary shape. So similarly, in this case, the substrate must have a complementary shape that will fit effectively into the shape of the active site. So we can say that Fisher's model is based on substrate specificity, meaning that there is only one type of substrate that can bind to the enzyme. For a very long time, this model was highly accepted, and in fact until today, to teach simple concepts about enzyme, we still use the lock and key model. But now I want to share with you why Koshland changed the hypothesis into the induced fit model. According to Koshland, he says that the substrate does not have to have a complementary shape to the shape of the active site. But even without a complementary shape, he says that the substrate will be able to bind with the enzyme to form an enzyme substrate complex, which eventually will form a product. How is this possible? Well, you see, in Koshland's time, our understanding of protein had improved. So as we know, enzymes are actually made of proteins, and proteins are flexible. So if proteins are flexible, the active site should also be flexible, unlike what Fisher said, where you see Fisher said active site should be rigid. But Koshland feels that because proteins are basis of enzymes, therefore the active site should be flexible and should be able to undergo conformational changes. So then, using this concept, let's see how the induced fit model works. Here once again, I have my protein enzyme, but to continue my discussion or for you to illustrate, this will be difficult to draw. So let me simplify the diagram to represent my enzyme. You can see the active site here is not rigid, but it is flexible. Now I introduce the substrate. As you can clearly see, the substrate structure is not in any way complementary to the active site. But you see what happens is when the substrate binds to the active site, the process of binding will induce. Okay, that's why the name induced fit. Okay, but induce here means like stimulate the enzyme's active site to experience a change in shape. We call this as conformational change. So once it changes shape, it becomes a perfect fit. Now you have your enzyme substrate complex, which will eventually form the product. So Koshland states that firstly, the active site of an enzyme is flexible since it's made up of proteins. Secondly, the substrates do not have to be complementary to the structure of the active site, meaning that he feels it is not only one type of substrate that can bind to an enzyme. Instead, he feels a number of different substrates can bind to the same enzyme. Only the condition being that the substrates, the different type of substrates, should have similar structure or similar chemical properties. So then they can fit into the enzyme's active site. Now, although the shape of the substrate is not complementary to the active site, but the binding will then stimulate the active site, the enzyme's active site, to undergo a structural change. Keep in mind that it is the enzyme's active site that experiences the change, not the substrate. Okay, then we find when it adjusts its shape, there is an induced change in the conformation of the active site, there will be a more effective interaction between the enzyme and the substrate, forming the enzyme substrate complex, which will eventually form a product. So Koshland's model's basis is that enzymes and substrates are group specific. So this concludes the discussion between Fisher's model and Koshland's model. I'd like to conclude this video with a small discussion of one past year question. In our STPM 2015 paper, there was an essay question on this topic where they ask you to describe the mechanism of an enzyme action based on induced fit hypothesis for 5 marks. So as usual, you know, if an essay is 5 marks, we try to write about 6 points. I'll share with you what I think should be the points added to your essay. Firstly, we should talk about the substrate. So we know that in the induced fit hypothesis, the substrate is not complementary to the structure of the active site. However, different types of substrates with similar structure or chemical properties will be able to bind to the active site of the enzyme. Second point will be, of course, about the active sites. So that when the substrate binds to the active site, since the active site is flexible, the binding will induce the active site to have a structural change. Third point, when the structural change happens, there will be an effective interaction between the enzyme and the substrate, enabling the formation of an enzyme-substrate complex. Once we have an enzyme-substrate complex, a biochemical reaction will be triggered. Product will be produced. Now, the product will exit from the active site and the enzyme will return its active site to the original structure. In conclusion, we can mention that the induced fit hypothesis is based on group specificity and introduced by Koshland. So by adding a little extra at each point, I am sure you would be able to get your full 5 marks. So that's all from me at BioWorld, wishing you the best with your STPM. Bye-bye.